Hello and welcome to another session and discussion of today's. So myself Pradit Banerjee, as you all know, and we are at Bio-ProTech. So in today's session and discussion, we are going to talk about molecular dynamic simulation. So it's a very important topic, and in the comment section, many of you have requested for the same, and we are going to see some snippets of it as this is a very long topic in order to discuss. So we would look at some of the use cases of it and how we can go about it and what is the difference between molecular dynamic simulation and docking. So in different stages and via different means, I would like to share the things. Okay. So first we'll have a whiteboard while like where I would be explaining about what is MDS, what is the difference between MDS and docking. And post that we'll look at the Gromacs tutorial and try to understand the commands related to it, how the commands like can be read, or what you can infer from all of those commands, and what you are basically writing in the commands, what you are instructing to the computer to pursue. Okay, and post that we'll also have a look at the result. Okay, so I already have a result of 20 nanosecond simulation. Okay, so I would show you that particular result using a visualization tool called VMD. Okay, so Visual Molecular Dynamics. So that is the agenda of today's discussion. And prior to moving on to all of those, let me discuss about what this particular thing is. So basically, molecular dynamic simulation is the ultimate validation parameter that you can think of in like your in silico. Okay, so when or in dry lab, you can also say so. Like in dry lab, we have this as a validation method, and you have, apart from this, you have your MMT PBSA, then you have your free energy perturbation. All of those things, okay, comes under this only. Okay, so we can use or utilize all of these to validate our result, because it's a question that comes every time to all of the students from biotech or life science or wet lab people, that you are doing all of the stuff in computer. How do you validate those? How you are so sure that all of the drugs that you are designing or the virtual screening that you are pursuing will actually land up well in the actual environment? Okay, or inside the body, how will it actually work? So in order to validate those things, like be it a drug molecule, be it a protein structure validation. Okay, so let's suppose you are coming up with a protein structure using your like any of the tools, like your AIML based tools like r.m.s.f or something of that sort. So how do you validate that particular structure? Okay, so it will actually work inside the body rightly, or how it will work. So in order to judge that, you need to do the validation of the same, and in that particular thing, so only molecular dynamic simulation can help you. What is simulation and all of that will be understanding in a while. Okay, prior to that, let me talking about the like analysis parameters. Okay, so there is no bar to the analysis that you can do in simulation, so there are n number of analysis possible. Okay, so but the major analysis that we do are of three types. One is r.m.s.d, r.m.s.f, then your radius of gyration. Apart from that, you have your like hydrogen bond analysis you can do, torsion angle, okay, phi psi angle. So a whole lot of analysis that you can do, depends upon what sort of a result you want to come up with, or what is your research question. Based on that, the analysis may vary a lot. Okay, so like there's a bigger scale under which you can do your analysis. Okay, so let's talk about the major three things or major three analysis that we have to do, and most of the time we generally do it. Okay, first is r.m.s.d. Okay, then we have r.m.s.f, and then we have rg, that is radius of gyration. So let's understand all of it. So first is r.m.s.d. r.m.s.d stands for your root mean square deviation. Okay, as the name suggests, root mean square deviation. So what we are trying to do is, so of course we have a graph, so x-axis and y-axis. Okay, so whenever we are plotting the graph, so what is this graph all about? First of all, what is r.m.s.d, r.m.s.f and rg parameter for? What are they judging? All of them are basically judging the stability of the protein. Okay, so how stable your protein is, or how stable how stable it is that you have configured. Okay, because we in the simulation, the main thing that we have, or the main difference to the docking we have in simulation, is the consideration of conformational changes. The docking does not take conformational changes into account, so you can think it of a snapshot or a picture, whereas MDS is a video of the same, that at what particular circumstances, because the protein would be experiencing so many things inside our body. Okay, so different temperature, pressure, okay, so electrolytes, whole lot of things are there. So under that particular circumstances, or under those particular circumstances, how the protein is performing and what is the stability of that particular macromolecule. Okay, so in order to judge that, and if let's suppose we are doing a like a simulation or docking simulation or protein ligand interaction simulation we are doing, so we need to see that how longer that drug molecule is inside that protein cavity or pocket. Okay, so that we need to of course judge for. After that we would come to the conclusion that what can be the implications of the same, or how long it is staying in that particular cavity or contour. Okay, so that is there. Yes, so basically r.m.s.d stands for root mean square deviation, as stated before. So it says that what are the changes that protein has experienced from its initial state to the final state. Okay, so what is the initial state? Of course, if you are doing a docking or a simulation, okay, so initial is the let's suppose the initial state we are talking about I0. Okay, so initial state that you have before the simulation and after the simulation, because let's suppose you are doing the simulation for 20 nanosecond or this that. Okay, so or let's suppose 100 nanosecond, that is the like standard. Okay, so for publication you would be bare minimum requiring 100 nanosecond. Okay, so after that you can of course go beyond that, 100 nanosecond, 200, 300, 400, likewise. Okay, so of course we can start with picosecond, but 1000 picosecond would be 1 nanosecond, and then 1000 nanosecond would be uh, like uh, 1 your millisecond, and likewise. Okay, so likewise, that we generally do picosecond to nanosecond, and 100 nanosecond being the standard. Okay, so beyond which you can like go for publication and all. So now coming on to the fact that what are the changes that the protein has experienced during its initial state to the final state, and in between of course the simulation has happened. So of course we are anticipating that very less changes should be there, because that means the protein is more stable. So the graph should be like not much of a fluctuation should be there in the graph, so it should be more or less stable. Okay, so it should not go like this or that. Okay, so or in between, let's suppose till 30 nanosecond it was very stable, and after that it went out of the box, or like it went out of the graph basically. Okay, so xy like x and z x and y graph we are basically plotting. Okay, so in that it should not go beyond the graph. Okay, so that is what we are looking for, that the graph should be by large stable. Okay, so and if we are comparing our protein also, so the wild type will obviously be having a very stable configuration or a stable graph that you will see, because it is a wild type. Okay, and if we are comparing it to a mutant protein and we are looking for a better stability of our mutant protein that we have modeled, so it should also follow that previous line or the wild type line basically. Okay, so that is what it gives, that nearer to zero, okay, or as close as it is to zero, better it is, because it defines the stability and there is not much of a fluctuation from its initial state to the final state, or any state in between. Okay, so the protein how we are taking at the initial state should land up as it is, or with some of the fluctuation at the final state. Okay, so final state. So that is what we are looking for. So that is why r.m.s.d should be as lower as possible, and as close to zero better it is. Okay, similar to that is r.m.s.f. r.m.s.f again should be again stable. Okay, so less of fluctuation should be there, but what we are judging for here is r.m.s.f first of all stands for root mean square fluctuation. Okay, so what it does, it also judges for the stability only, but it judges for the stability at the amino acid level. This like r.m.s.f judges at each at amino acid level, because of course the protein is made up of amino acid only. So whenever we are talking about r.m.s.d, we are talking about the whole protein. Okay, the full protein. Okay, so the full 3D structure that it has acquired. Okay, so be it a secondary structure, tertiary structure, quaternary structure, whatever it is. So the full protein we are considering. Whereas whenever we are talking about the r.m.s.f, we are talking about the individual residue that it is made up of. Of course, that those are the amino acid, because the 3D structure that is there is made up of amino acid, of course. Okay, so we are judging for the amino acid, same thing. r.m.s.f would be the same thing, that what is the difference between the initial state to the final state, but at residue level. Okay, so at each residue level. So that is why the graph that you plot over here, okay, so would be against residues. Okay, so like the previous one, the r.m.s.d would be against your time. Okay, so against your time, the like in how much of a time, how much of a fluctuation is being seen in the protein, that is your r.m.s.d. Whereas in the r.m.s.f, it would be against residues, that like at what particular residue or what particular number residue caused the fluctuation. Let's suppose I was taking the example that after 30 nanosecond there was a peak that you have seen. Okay, so that particular fluctuation has happened because of which amino acid, okay, or what particular amino acid conformational change has caused that particular fluctuation or deviation of the protein or destabilization of the protein, that you can understand via r.m.s.f. Okay, after that comes your rg. Okay, rg, capital R, small g, it is represented by, that is called as your radius of gyration. So what is that? You have the whole protein. Okay, because in because in your MD simulation, what happening is, so there is conformational changes. Okay, so conformational changes means what? As the thing is in a 3D space, so it is not only revolving about its axis. Okay, so it can do it along the axis and about its axis. Okay, so it can like rotate or change its conformation in multiple ways. So that is why what we judge in a radius of gyration, as the name suggests, radius. So we are taking it from the center. We are taking it from the center of the protein, then we are judging for the radius, half of it, of course. Okay, so half of it would be the radius, and that particular radius would be the revolving part of the protein. Okay, so how much it is like revolving. Okay, so this way or this way. Okay, so both the ways, of course, it can revolve or change the conformation. So the half of it, that is the radius that we are judging for. So here also we are looking for the stability, that rg should be like that, and radius of gyration also gives us the stability, but not in the fashion of your whole protein or residue basis. It is judging for the whole protein, but assessing like how horizontal rotation or vertical rotation it can have. Okay, so it is based on the spin or rotation of that particular protein inside our body. Okay, so that is about all of the three parameters judging for the stability, but in very different manner. Okay, because all of these things are possible, so that is why we are judging at the whole protein level, residue level, and the spin level. Okay, so that is why that would give us a clearer picture that what are the conformational changes that are happening, and because of which, if there is a drug or a protein interaction that is happening, how long it will stay in that particular thing. Because if the conformation changes, let's suppose this was the drug molecule in the protein cavity first, so it would be interacting with different amino acid first, and if it changes its position, now it is exposed to different amino acid now, so the interaction would be different. Okay, so that is why we also do an analysis of hydrogen bond, that during the whole course of simulation, how much of hydrogen bonds basically build up or broke. Okay, because this may happen, because let's suppose in this docking, you have seen that 11 interactions were happening, but that is just a snapshot. Okay, so in the total 20 nanoseconds, like everything is basically levitating or revolving around this axis and all. Okay, so it would be changing conformation, because of which it would be nearer to or closer to different amino acid at different times, the chemical entity, the ligand. Okay, so that is why it would be bridging a different sort of a bond. Okay, so it may be hydrogen covalent or like all different kind of interaction that it may have, that would tell us that the strength of the interaction for a longer period of time. And of course, how much longer we can run the simulation for, more the merrier, of course. Okay, so that is why with the advent of newer, faster computational system, okay, like the supercomputer, quantum computer, and likewise, we can move up to your millisecond simulation, and that would be more fruitful. And if we can do it for hours simulation, okay, so like by the virtue of your quantum computers and all, if we can reach hour simulation, so that would be great. Okay, because more amount of time if we can simulate a particular thing for, okay, so we have a clearer picture of for in the longer period of time what that particular interaction would look like, and that would be exact thing that will happen inside the body, and that of course can be validated via wet lab experiments. Okay, so if you are thinking that we do not revalidate or cross validate it using wet lab experiment, no, we do. Okay, but after this is being done in the computers, of course it is far easier, cheaper, faster. Okay, so that is why most of the pharmaceutical companies first in the initial phase does this particular thing, because let's suppose we are boiling down from 10 lakh drug entities, or leads basically. Okay, so initially it would be called as leads, so we are boiling down to 2 or 3, then we are like finalizing those using docking, and 2 or 3 we are doing a simulation of, and when we have finalized that it will actually work, then we validate it using your in vitro, in vivo studies, then the clinical phase trial 1 2 3, and then post market. And likewise the approval committees, like the USFDA would be approving in US, then EMA would be approving in like European continents and subcontinents, and likewise in India you have Cedisco approving that particular drug, and after that the post market what do you say, observation starts, because once the drug is marketed also, after that also you do observe it for a longer period of time. Okay, so that is there, and that is how a drug is being made nowadays. Okay, so with the help of your computer aided drug designing, the virtual screening, then your docking and the simulation, all of these things are made very like have made this particular pipeline very very easy to understand and to screen in a faster pace, and saves a whole lot of money, of course. Okay, so that is why we are having this particular thing. Okay, so that is that, and let's move on to the further part of the things with the help of the whiteboard. Thank you. So here we are, basically we are talking about molecular dynamics simulation. Okay, so this particular thing can be used in multiple cases actually, or like it has multiple use cases. So what are those? First things first, whenever you are like looking for a like protein ligand interaction, okay, so you are going for a drug discovery of that sort. Okay, so whatever it be, so the final step of validation in your in silico based experiment is MDS. Okay, in any kind of in silico based experiment dealing with your like in vitro, well what do you say, your drug discovery that you're talking about. Okay, in order to validate any of the in silico based results, okay, you will be requiring this particular thing as a validating parameter. So why is it a validating parameter? So we'll talk about that. Let's suppose if you on the drug discovery pipeline, so you just start with let's suppose 1 lakh compounds, 1 lakh odd compounds, then you do the screening of those. Okay, so using your drug likeness. Okay, where comes your Lipinski's, Pfizer rule, this, that, okay, a whole lot of Weber rule, Ghosh rule. Okay, then you do your pharmacokinetic and pharmacodynamic T-based screening, that is called as a ADMET. Okay, so ADME oblique T. Okay, ADME falls under your pharmacokinetics, whereas toxicity falls under your pharmacodynamics basically. So basically ADME falls under your PK, that is what I said, and T falls under your pharmacodynamics. Okay, PD. So what does it signify? Basically, like your pharmacokinetics focuses on like what body does to the drug. Okay, so this is basically body's response to the drug. So that is why absorption, distribution, metabolism, excretion, all of the things are carried out by our body, organs in our body. Okay, whereas toxicity is basically what drug does to the body, because toxicity won't be produced inside our body. Obviously, the like our organs won't produce our own toxicity, isn't it? So the external molecule, that is drug in this particular case, which is being administered, so that might be having a toxicity profile. Okay,
and that particular thing will be studied upon. Okay, so how much toxic it is, or likewise, okay, so is it having a bad effect on your liver or your kidney or your any of the important organs out there. Okay, based on that you screen, and at the end, let's suppose you are getting like 20 or like 20 to 50 odd compounds which passes all of the filters. Okay, or even more than that, in hundreds also you can get. Okay, depending upon what is the screening parameter, how stringent it is. Okay, based on that, after that, let's suppose you do docking. Okay, so docking is what? Docking is basically knowing the interaction between the ligand and the protein. So this is a ligand, let's suppose, which will be called as a drug molecule later on, and then let's suppose this is the protein. Okay, so in any contour it might fit. Okay, so you have to look at the docking for that matter. So basically docking softwares will try to fit this particular ligand to any of the contours, be it your pockets or cavities, where we are seeing the how the ligand molecule is interacting with the protein molecule or the macromolecule. Okay, but this particular thing is happening in a 3D space. Okay, obviously it is also 3D, but it is in an inert environment. Okay, so in an inert environment, it has nothing to do with your actual physical environment that the drug will actually experience inside the body. So that is the main question that is always asked, that you are designing a drug in the computer, how will it work in the humans actually? In order to know that, in order to get a practical aspect of that, so for that particular thing we do a simulation. Okay, so this is where we built the relation why the simulation is required in the first place. Okay, now coming on to the second question, that like why we do so. Okay, so and how we do so. Okay, so that is the whole session of today's is about. Okay, and the main thing is like what is the difference? Simulation anyways means to mimic. Okay, in any prospect it means the same, that to mimic. So let's suppose you are like working on a building. Let's suppose we're talking about Burj Khalifa when it was made. Okay, so obviously add like if you are building a thing of that stature. Okay, so that would be experiencing so much of winds and this and that thing at the top. Okay, so it has to be simulated. Okay, it has to be simulated. The similar conditions are to be simulated, the desert storms, this that, how it will experience, and how it is it is so much flexible that it can withstand that or not, because you cannot like directly work upon that multi-million what a billion dollars project directly and then see, isn't it? So you will be having a pilot scale of that particular thing. Okay, tabletop or pilot scale of that particular thing, and try to simulate the exact conditions and see how it works, the exact thing that we do over here. Okay, instead of going with the drug directly for your in vitro in vivo studies, and after that obviously you'll go for your clinical studies and all. But when we are following the whole in silico pipeline, whatever I have told you in here is called as a CADD, computer aided drug designing. So that is your whole in silico based method. Okay, so by this via this particular means you can save a whole lot of money. Not like all of the pharmaceutical industries, you can read papers about it, be it your Pfizer, GSK, or your um, Novartis, or whichever company you take. Okay, so they have saved a whole lot of money using this particular pipeline out there. Okay, and before administering the drug, or even for going for the clinical trials or in vivo studies, it is obviously better to study it in the computers by simulating the exact human conditions. So what is the requirement of that? The main thing is that the drug, while getting inside the human body, would be experiencing multiple things, because the protein that you see is will not be like in a static state. Okay, so inside the human body, because it is experiencing a whole lot of things, like our body is 70% water, then it has multiple electrolytes, so obviously that is having charges. Okay, so positive negative charges, like your sodium is there like positive, then your chlorine is there negative. Okay, so those things are there. Apart from that, our body maintains a certain temperature, it works in a certain pressure. Okay, so those all of those things are to be mimicked. Okay, so that we can come up with a condition that in this particular condition, so obviously inside a box we will set up all of those conditions, and in that particular condition how this particular two things are interacting for how long. That is what we are trying to see. For that, we have multiple time tangents also. Okay, from your picoseconds to nanoseconds to millisecond. Okay, so that is there. So how much time it can hold on to that particular what do you say, interaction, okay, in the actual what do you say, space, while it is experiencing all of the temperature changes, this, that. Even at atomic level, there are multiple changes. Like when I show you the results at the end, okay, so I will show you a demonstration of the simulation result. There you will see all of the atoms are basically levitating, or basically revolving around its axis, as well as along the axis also it can rotate. Okay, so that is why it will seem vibrating, but it is actually not. It is actually levitating, or like rotating about its axis, and likewise. Okay, so then we can come up with multiple of your like evaluationary parameters that are there in MDS. The results can be analyzed in multiple ways, because there is no restriction to your result analysis over here. Okay, so you want to come up with your r.m.s.d. Okay, so r.m.s.d is there, r.m.s.f is there. Okay, so I will talk about all of these things, okay, in the session. Okay, so r.m.s.d, what is r.m.s.d, what is r.m.s.f, what is rg, that is your radius of gyration. Okay, rg, uh, r and small g. Then there are many things like your hydrogen bond analysis, torsion angles. Okay, so at atomic level, every detailed studies you can study you can do. And MDS is also not of only one kind. This is what I was giving you an example of protein and the ligand. Okay, so the drug molecule and your receptor basically. Okay, but it can work with only protein, that is called as your protein in water simulation. It can like give you this particular interaction, protein ligand simulation, it is called as. Apart from that, free energy perturbation is there. Then multiple types of assays are there. Okay, so likewise. Okay, so so that is there. Okay, so I will just give you so this is just the overview of the things that we are going to cover in today's session, and just to establish the relationship between why we are studying it, how is it relevant. Okay, so likewise, Gromacs tutorial. Okay, so this is your regular search engine that you'll be using, and there you need to type Gromacs. Okay, this is the spelling of Gromacs. Okay, tutorial. Okay, go to the first one, mdtutorial.com gmx. Okay, so here you will see what types of simulation it can do, mostly 7. Okay, this is the free software by the way. Okay, so you can see what are the modules. Each of the modules is being described over here. Okay, so basically this is the lysozyme in water is the most basic, the protein in water that we are talking about. Even this is updated in this particular year. So likewise, then you protein ligand complex for the drug molecule, free energies of solvation or perturbation. This is there. Multiple by physics solvents that you want to simulate. That is also there, like the oil and water, this, that, or oil and some other fats that you want to simulate. That is also possible. Okay, so this thing you can do. Then your umbrella sampling, multiple things are there. Just you need to understand from a biologist perspective, or a like computational biologist perspective, or your chemistry perspective. These two are mainly important. Okay, so this is just the same thing that you have to do it for protein and ligand both, and then pursue. So I will show you via the first one. So you just have to go inside this, start the tutorial. Each steps are mentioned very, what is a stepwise. All the like what is a codes, not these are not codes, basically these are commands. Okay, so basically Linux or Windows that you are using, these are basic commands. Okay, so if you know the Linux commands, mostly you will understand these things. Okay, it is very simple to understand. If you're not understanding anything, everything is described. Okay, you can take a help of that. So that is why I have shown you this particular step, so that you can learn at your own pace. Okay, so basically first is gmx module, is the Gromacs module. Okay, so you need to install that. Okay, so after installation, obviously grep grep basically. So grep is the command, so where you adding water to it. Okay, so like removing water, pardon. Okay, so minus v, HOH is H2O, basically removing water and all the heteroatoms, because for docking also we need to remove the heteroatoms. For your simulation also we need to remove the heteroatoms. So this is called as pre-processing of the protein, where you remove the heteroatoms and add the missing residues. Okay, so that is an absolute basic thing for all your simulation docking, everything. Okay, what is that? What is a heteroatom? Heteroatom is anything which is not a protein. Okay, so which is not a protein means anything which is not required by the protein to function solely. Okay, if it is a pre-attached ligand or like any water molecule attached to it, any metallic ion attached to it, which is not useful for the protein. Okay, so you might get those things in PDB because that was the experimental requirement, but now you don't require that because for your experiment you need the raw protein. Otherwise, the heteroatom will hinder with your like simulatory result as well as your docking based results likewise. Okay, so here we are basically removing the HOH, saving is at as a PDB. Okay, so you can see clean.pdb, it's a new file name. Okay, likewise, then this excuse me, yeah, so then you have to process it after cleaning. Okay, so all of the things can be done, and then you add the force field. These are the choice of the force field, you can choose either of them. You can see amber, charm, Gromacs, OPLS, that is what I said. Okay, so then adding topology, I have already told you. Next step is to like generate the topology file, and every time the topology file is to be called because everything is to be updated in the topology file. Okay, so include generate topology file. The charm is being selected over here. Okay, then your yeah, so then be done. After that you have like the compounds and then adding the solvation. Basically first we add the box first because inside that the solvation would be added. So here you can see. Okay, so F is the function, GMX is Gromacs. Okay, so O is what should be the output. Okay, so so basically this is the file that it is asking it to take, and this is what it should give as an output. That is the command all about. Okay, then what to do, this is the name of the like code of the box, and minus bt cube, cubic box we are adding. Basically you can see 71% of the cubic box volume is being used for the periodic distances. Okay, so after that the solvation is added. Okay, so after the solvation is added, so this is this SPC 216 is a code basically for your water. Okay, so new box .gro, this is the code for your water which is added. Okay, so then update in the topology file. Always you will write this particular step, that whatever step you are doing, update in the topology file. Then only it can keep a check of all of the things. So you can see protein chain it has found one, and the like solvent added, basically water that is added is this many molecules of waters are added. Okay, then adding ions to it. So you can see the ions. Okay, so adding ions, ion.tpr file. Okay, so that would be added. Topol.top always update the topology file. Okay, so obviously you can see why we are updating, because we are always changing the name because of the file. Otherwise, we will not able to relate that what is being added and not. Okay, in one file, if you are adding all of the steps, so that is not the right way to do. Okay, then after that, you just write in Na and Cl and neutral. Okay, so what does it mean? So it will calculate that whatever is in heavier quantity, the other thing will be taken like given to it to neutralize. Basically, if Na is more, Cl will be given. If Cl is more, Na will be given to neutralize it. Okay, so here you can see 8 molecules of Cl, chlorine are added. So basically, it was higher on the positive side. Na was high, so that is why Cl minus is added to neutralize it. Then coming on to energy minimization. Okay, so in the energy minimization, you basically add do the energy minimization step. Okay, so you see the potential energy minimized. Okay, so the graph is also there. Here you can see the graph is falling. So obviously this is the step where the energy is being minimized. 500 something step. You can 575 or something would be there. Okay, yeah. So now after that comes the equilibration step where you will be having the NVT, NPT. Okay, so NVT like volume temperature constant. Okay, here you can see this is the like actual graph in picoseconds, so 100 picosecond graph. So obviously it will, you can increase based on how much time you are doing it. Okay, so similar graph will come for your NPT as well. Okay, so you can see the pressure temperature graph. Okay, so as closer it is to zero, obviously it is better. So that is the central line as you can see. So this is for the wild type mostly. Okay, so the density also you can see, density again, it should be closer to a this particular central line it should follow. Not a straight line, obviously it won't have straight line anyway, because this is a simulation. Okay, So at every step it is taking, but the closer it is to that, better it is. Okay, so yeah. So after that, obviously, MDS 0th step will be taken. If you have the GPUs, you can write this particular command as MD run, then MD 0 10. Okay, 10 means it is not that you always have to write 10. 10 is for the 10 nanosecond. Okay, if you're doing for 100 nanosecond, you write it likewise. Okay, so it is just for the understanding sake. Okay, then coming on to analysis, after that comes the analysis part. Okay, after that, obviously your frame rate and that particular video is also created. After that comes your, like, you can do the central of that and go to the analysis part. The different type of analysis that you can get. Okay, so this is the radius of gyration. Okay, again central line, better it is. Okay, so this is the density secondary structure plot. Okay, hydrogen bond plot, how many hydrogen bonds are formed, and the side chain, the main chain that we need to see in the protein water simulation, what it is like, what should be the ideal condition, that is what is given. Okay, so for you, obviously this particular thing will not be that straight. Obviously you will be having some ups and downs. Likewise, okay, so likewise you can just see the whole thing. Everything is detailedly described over here. Okay, so likewise. Okay, so last but not the least, so let's take an example and see that how the particular thing looks like. Okay, so I will just come on to use your VMD for this particular purpose. Okay, so VMD opens in command line basically. So in the background, this particular thing will be open. Okay, so and you have three setups out here. So this is the visualizer tool. Okay, so this is the main thing from where you'll be operating. So you'll go to file, new molecule. I have already a file. So this is the secondary browser. You can see. Okay, so not browser. Yeah, so this particular browser can be used for browsing. So you can go to basically browse. Okay, so then I will select your, so let me get in this over here so that you can understand. Okay, so I can go on to your, I know it is in downloads. Okay, so I will go to downloads and yeah, so I can apply custom filter because there are a whole lot of things to find. It will be easier. So first thing that we will be searching for a .gro file because the simulation is done using Gromacs. So it will be in a Gromacs file format, .gro. Okay, so search for that. You can see MD020.gro means 20 nanosecond simulation it is. Okay. Okay, if I do it and hit on load, you can see the Gromacs file. Here you can see a protein with all of the water molecule and a box is loaded over here. Okay. So if I stretch it, I will zoom it and I will do it full screen while I'm doing it. Let me, what do you say, load the frame rates. So based on what you can see the movie part. Okay, so that is called as an XTC file. Okay, so that is um, compressed file. So just give me once again, .xtc. Okay, if I search via that particular custom filter, so here it is. Okay, so now here you can see. Okay, so you can see everything levitating, every molecule is rotating. In between, you can see a blue mark. Okay, so that is basically what is happening over here. So that particular protein is, now you can see it is getting out of the box. You can see at the end, it is getting out of the box. That signifies that we need to run the simulation for a longer period of time and see that it is getting out of the box for a moment, but is it coming back or it is going out or it is being destabilized? Those things are to be, so likewise we judge basically. Okay, so I will try to run it like once again and do it a full screen so to make you understand. Here you can see. Okay, so it is obviously in between as of now. Okay, so I will like zoom out and zoom into so that you can see this one. Okay, so the particular cavity is left because the protein is going here and there. Okay, so that is the way. So obviously, likewise you do the simulation for one after another. So yeah, that much I have stored in here. Apart from that, multiple hundred or more than that we have done, but that particular results are not with me. This is there, so that is what I'm showing you. Okay. So basically, to just to give you an idea how the things works. Okay. So that is there. Okay, you can see all the frame rates here. You can see 1600, 1700 something. Okay, current frame rate. So this is the frame rate, how fast it is going. That is why you can see it in the form of a movie, but this is an animation. Okay, so you can also change the step, or you can increase the speed. It is at the maximum speed. If I just slower the speed or the pace, here you can see it is going frame by frame, very slower frame rate. Okay, if I increase the frame rate, then you can see all of this moving very fast. Okay. Likewise, so this is called as your trajectory file, basically. That the second one that I have loaded, XTC file, it is called as a trajectory file, the thing we were talking about. Okay.
Yes, folks. So that is all from my side as of today. If you have found this particular session engaging and knowledge worthy, so consider liking and following this particular page. And I'm really very sorry that I am not able to post very frequent videos under this particular channel. Okay, so because of my multiple engagements that I have, okay, and I am also on to multiple of the projects and my scientific work that I'm on to. Okay, so I am very sorry regarding the same, but I would try my level best to post more frequent videos in the upcoming future. And with that note, you can also consider looking back to all of the videos that are posted lately or like previously in this particular channel. You can also consider going back to the docking videos in order to understand what is a molecular docking and how to pursue that. And after that, you can have a look at this particular video once again to understand what are the differences between the docking and the simulation. So with that note, so this is Pradit Banerjee signing off. See you in the next one. Till then, take care. Bye bye. Good night.